It's a really great pleasure to get to introduce Tanya Berger-Wolf, who's joining us today from The Ohio State University, as I've been corrected many times, where she recently took a position as the director for the Translational Data Analytics Institute, as well as a professor in the Department of Computer Science and Engineering. And she had the dubious pleasure, I'm not sure what to call it, of taking that job just in time for COVID to start spreading across the world and get to take over the university's rapid modeling response team, working to provide predictive computer modeling of the spread of COVID-19 to the Ohio Department of Health. So that has been taking up an inordinate amount of her time in the last year. And so I should, anyway, anyone who's feeling in any way insecure about their productivity as a scientist, or maybe even as a human being, should probably avoid looking at Tanya's CV. I may have done irreparable damage to my psychological well-being earlier this morning, but Tanya really is a force of nature. And she's worked across numerous fields at the intersection of computer science, ecology, population biology, neurobiology, starting out with a bachelor's of computer science and math, double major at Hebrew University in Jerusalem, and then traveling to the US to the University of Illinois, Champaign-Urbana, where she did a PhD in computer science, looking at multi-channel communication and graph vertex labeling. She did a number of postdocs, one on phylogeny reconstruction at the University of New Mexico, and a second on computational epidemiology and population biology at the Center for Discrete Mathematics and Theoretical Computer Science, before landing at the University of Illinois, Chicago, as an assistant professor, earning tenure, and then being granted, earning full professorship in 2016. So in that time, her research has really focused on developing and applying computational methods to answer a really wide and diverse set of questions about ecology, population biology, and the population biology of plants and animals, including us, humans. And she's collaborated across a really wide range of disciplines. And one of the things that really makes her stand out as a computer scientist, I think, is that she actually gets out into the field and gets her students out into the field as well. So she's really done amazing things, bringing ecologists and computer science together in field environments. And she's done that despite her insect phobia. You should hear the noise she makes when a cockroach crawls up her legs. It's spectacular. But all of this has really resulted in an amazing contribution to the scientific literature with over 100 publications. I stopped counting at $15 million in competitive grant funding, 13 PhD students, 34 master's students. But I think that what really makes Tanya stand out is that she's a really generous collaborator and mentor. She certainly taught me a lot of what I know about sort of the nuts and bolts of how to be an academic scientist. And I wouldn't be here without her. So Tanya, thank you. And the last thing that I want to say about her before she starts her talk today is that she has made wide contributions, not just in science, but in applying science in the real world. And so I'm sure that she'll talk about some of this today, but she's made significant contributions to the field of wildlife conservation, as well as playing a really important and visible role in promoting women in computer scientists, really from the early stages. So one of the joys of working with Tanya is also actually getting to work with the young high school and college age scientists that Tanya brings to our projects. So Tanya, we're excited to hear about your work.
Thank you, Meg. This is quite an introduction. I kept on looking around, like trying to find the person you were introducing. But it is truly delightful. It's a special moment because I've known many people in the audience, and Meg, I've known Meg since that postdoc. Well, not mine, yours, at Princeton. So. Because even though my official postdoc in DIMAX was in population biology and epidemiology, that was kind of the way to, we found a way to pay for my postdoc with Simon Levine in Princeton. So that ended up leading to interacting with Dan Rubinstein and his students, Shiva Sundaresan and India Fishoff, and talking about zebras. And they dragged me out to the field, and that was kind of the beginning of this quest of figuring out how computational methods can help understand social behavior and decision making of animals other than humans. And I really appreciate fieldwork and making my students appreciate fieldwork. And so with that, I'll tell you a little bit more about it. And as I said, there's so many people in the audience that I've interacted over the years. So and it's great to see these names. Hi, Martin. the one thing I want to ask is that as I'm giving the talk, it's very hard for me to keep track of any kinds of questions, raised hands, or notes in the chat. So if there is a question, please stop, because the talk kind of has some reasonably self-contained parts. So please ask questions. Don't wait till the end. Just pipe up. I'm normally a very interactive speaker with audience participation. So if in this format it doesn't quite work as well, make it so. Just talk. There will be quizzes. All right. So with that, let's get to it. So the work that I'm going to present, a lot of it was done at University of Illinois, Chicago. So they get full recognition for this, and the Ohio State University. More recently, as well as Wild Me, which is the nonprofit I'm a director of, and Wild Book, the project of this nonprofit, Wild Me. I also want to give recognition to the other two departments that I'm a professor of at OSU. So I have a multiple research personality. So that's electric computer engineering, and for the first time in my life, officially, evolution, ecology, and organismal biology. So with that, you know, the scientific method, the way we study natural phenomena, the approach has been set out by Poincaré, and there is a wonderful, wonderful little book that is called Science and Method, from where we really draw our, this whole notion of observation, experiment, hypothesis. And he ends this book with a statement that the scientific method consists in observation and experiment. If the scientist had an infinity at his disposal, and it is his at the time, of course, it would be sufficient to say to him, look and look carefully. But since he has not time to look at everything, and above all, to look carefully, and since it is better not to look at all than to look carelessly, I think I constantly tell my students, he's forced to make a selection. And the first question then is to know how to make this selection. And so in this process, what does computation really bring in, or data science really bring into this process? Because with this description, it's not that science, data science and computation suddenly drastically changed what science actually is about. It hasn't. Science is fundamentally data science, all sciences. So what computation brings to this process of asking a question and getting to the answer, which scientifically is typically the answer that starts with because, right? Because the scientific questions are the why questions that go through this process of observations, data, some intuition, logic analysis that lead to hypothesis, with a cycle of going back to more data, more observations, and more experiments, and at the end, hopefully a plausible explanation and a supported hypothesis. Computation, in my opinion, and technology bring in this expansion of the range of data, the type of data, the quantity of data, the resolution of data, the detail. They bring in the ability to extract different patterns, make essentially different observations on that data, and ultimately, hopefully, a way to translate these observations, patterns, and relationships into something that resembles a hypothesis. Because the models that come out of computational approaches are not explanatory models. They do not explain scientific phenomena. They're great predictive and descriptive models, and but the partnership has to be there between the scientists and computer scientists and computational approaches to really lead from these observed patterns to scientific hypotheses and explanations. And so what I would claim, what I would propose, that really what computation and technology brings into the scientific process is the ability to look at more things more carefully and hopefully answer questions, ask different questions, and answer um, the questions that we weren't able to answer before. And so with that, the particular kind of part of this scientific process that I've been focused for the last 16, 17 years or so is really the part of social behavior and social decision making. And so this process where we, and to borrow this is thanks, part of it is thanks to Peter Holm, the description. So we start with data, where a lot of what we do in this domain is really we model as networks. And so we define these networks, and we'll talk a little bit about that. Then we represent it in some abstract way. And when the network is dynamic, there are many options. Go to this process of extracting patterns and structure, which then lead application insight and through scientific hypotheses. And the connection between the science and the computational abstractions and the computational analysis really strongest when between translating from data to the abstract definitions and leading back after the extracting patterns and scientific and computational relationships to scientific insight and hypotheses. And so I'll some examples, pretty high level examples of this process me at any point. So I don't think this would look unfamiliar that data in ecology comes from the field, and our data collection process often, and in social, for the most part, pre-technology or without technology and compute, there's a scientist, and in this case, David Papano, former post, who then either scans the entire population, gelada, not ice cream for computer scientists. This is not ice cream, and these are not baboons, I'm told. But they are close relatives and completely herbivore primates, and are very easy to habituate to humans. So they're easy to observe, and they're you can stand there and just watch wild animals do the thing they do. But the thing is, even with whether you're focused on one animal and try to write down everything that that animal does or do the scans of the population, it's very hard to observe every animal in the population at all times. And you notice perhaps that they're in the back on the top left. There are gelada that probably none of those three observers actually are paying attention to at any given moment. And so the thing is, this is the fact that we're missing data about the social interactions, but we quite often don't even have the basic data of how many of these animals there are. And on a global scale, so if we talk, the International Unit for Conservation of Nature that is supposed to track the population status of the most endangered species and determines the endangered status of species. So when we say that the species are endangered, it's because the IUCN Red List Species Commission determined that they're endangered. So they right now track more than 130,000 species, and out of millions that are estimated to be out there. And of those, more than 16,000 official status is data deficient, not something obscure, killer whales, right? So if we don't even know the population size of these iconic species, then we don't, the basic data for the rest is kind of, seems hopeless. for many, we don't know the population trend. Is it going up, down? And that's because they're not observed often enough. So polar bears and other iconic species. And when we do know, so for example, whale shark, the biggest fish on Earth, in 2009, the global population size of the species was estimated to be 103,000 with a standard error between 27,000 and 180,000. That's a very scientific way of saying we have no clue, right? So how do we get a clue? One way to find, so today, images are the most abundant and readily available source of information about pretty much anything, from what you had for lunch to what animals are there. And in fact, for the most part, this is what we do when we go out and watch, right? Images are another way of just looking more carefully, hopefully, if we can extract information out of these images. And by the way, these are pictures from a course that Dan Rubinstein and I and Ian, I haven't seen Ian in the audience, but I'm sure he's there. So yeah, you're here. So Ian was was part of this experiment where we took computer scientists and ecology students, gave them some basic introduction to both disciplines, and threw them out in the field in Kenya and told them to do something interdisciplinary. And they did 3 times. We ran this course, and 3 times amazing things did come out of this. And so the top, well, this is the first time we taught this course, and this is a computer science student from University of Illinois Chicago on the left, a ecology, evolution, and biology student from Princeton, and a student from natural resources program, so all collaborating. In this case, observing, they were trying to understand bomodynamics. So the thing is with images, we need to be able to extract information out of them. So not only to look, but to look carefully. So and be able to take this millions, actually billions of scientists, field assistants, camera traps, drones, as well as tourists taking their safaris and whale watching tours and media, and figure out where they are. And so that's the project was started into 2010 by Mike Lahiri, the first iteration of this field ecology class, identifying individual zebras from photographs, and grew to this the project called Wild Book that Dan Rubinstein, Jason Holmberg, Chuck Stewart, and I co-founded, of building a platform where, using machine learning, we can take all the images that are out there, find the ones in them, identify where the animals are in those pictures, so include calf elephant hiding behind mom, or the self-obscuring zebras, and figure out not only down to individual animal, as in Zippy the zebra, Joe the giraffe, Terry the turtle.
Amazing amount of photos. I wonder, how do they sort through them all and then identify individual animals? Each image is processed through a series of convolutional neural networks and matching algorithms. This network determines whether there are any animals of interest, in this case, zebras. The next analyzes each animal in its own subregion, species. At this point, is performed on the subregion to generate a zebra. Once this points are extracted from the zebra and featured, these descriptors serve as a sort of fingerprint that can be compared for similarity against previously animals in a large database. Comparison allow the system to decide which animals are the most likely matches. When the match has high enough confidence, it's accepted. Matches that have lower confidence, expert, who then makes the final decision.
And we can do it for any stripe-spotted, wrinkle-notched animal, including also of different algorithms, including using the fluke of a whale, the trailing edge, or the dorsal fin of a dolphin. With information on when and where the image was taken, you can now use images to track animals, count them, even identify population dynamics, birth, deaths, as well as their social network. So here at the bottom of the, here's an example of for whales and dolphins for cetaceans. And Pinchy, our favorite sperm whale, she's a celebrity. She's animal among those. And they are social animals, so you can see the extracted automatically from all the sightings, the social network of this particular. She hangs out around Dominica. Today, Wild Book for whales and dolphins, millions of images, is actually more than 1.2, with more than 45 individuals. We also built a intelligent agent that can scrape posted photos on social media. In this case, an example of a YouTube video of a whale shark identifies the species of interest, finds sends it off for identification, uses the title and description to the video was taken, and with that, adding it automatically to Wildbook, posting them back in the comments saying, hey, at 2 minutes, 46 seconds, we found this whale shark that you, and here's everything we know about it. And people get excited because they didn't even know that their vacation videos are going to be used for science and conservation. Most people respond back, and the most common response is, wow, this is amazing, how can I help? So that is, immediately engaging people. But there's and growing this network and of people that are engaged in conservation, but it also grows the network of scientists, because at the bottom of every page of an animal, there are all the science and conservation projects that have that specific individual in their data set. And so particularly for global species like whale sharks, there is not one project, not one individual or conservation organization that have information even about one individual completely. So you really need to combine that information. And so today we have, for whale sharks, remember that those species that we had scientifically no clue of how many. So we have more than 12,000 identified individuals with information coming from almost 9,000 citizen science contributions, more than 200 projects, research and conservation projects, and one very intelligent agent that actually today brings more than half of all the sightings. So it brings the social media postings contribute together more than all the human contributors combined. So that's a blessing and a curse. And so the entry for IUCN Red List has been changed based on the information from Wild Book, and in fact, the conservation status of the species has been changed from vulnerable to endangered, because not because the species are doing worse, but because we now have data. The most comprehensive study on the biology of whale sharks was co-authored by 37 authors that met through the pages of Wild Book and based on Wild Book data. We have Wild Books for 53 species and growing, spanning marine to terrestrial all over the globe, using 7 identification algorithms and increasing. So we're looking to add primates that I know is some work is being done at Max Planck, and also Damien, we're happy to identify birds and add birds to wild bugs. And in fact, we've been asked repeatedly of connecting wild book to eBird, so let's do it. There are more than 80 that we know of publications that used wild book data for a variety of species, and every day we now get pings. But the problem with using, as I said, using social media data and in general using observation, using photograph data from this variety of sources, is that there are many biases in translating that data into actual scientific estimates of any kind, including the very basic estimates of population size. So, you know, you have the true population out there. The very first bias of being at the right place at the right time to even take a picture, to see it, right? Whether it's a camera trap or a human on a safari, there is the spatial temporal bias, and it's been well described in literature, starting with camera traps and drones and other um, observation modalities. The next one is the capturing that image, the bias of deciding to take a picture. So if you go out in the field with Damien, and look at his SD card, majority of the images you'll see there probably are going to be birds. If you go on a safari tour with somebody for whom that's the first one, you'll see a lot of impalas at the beginning and zebras, and then it starts tapering off. So there are people that take pictures, decide to take pictures of different things in different animals. And then there's the bias of posting it on social media. Which social media, what are the ways of collecting information from that social media, how we then model the population size from that. And finally, all of this has to come down to minimizing the error between our estimate in the original population and the true population size. And the thing is, it's not by figuring out the bias at every stage that we're going to get there, because these biases are also dynamic. And so how do you do, and what's now is termed pragmatic machine learning, how do you do this end-to-end task-focused minimization? And I'm happy to talk about it quite a bit. And a lot of the work that we're doing right now is focused on different aspects of minimizing bias or estimating bias, of figuring out how to estimate population size from social media, and what else can we get from social media, including work by most recently, and watch the space, most recently a team of 14 high school and undergraduate students who've collected data from 4 different social media platforms for 6 species, and analyzing it for different distributions and properties of how people use social media for wildlife animal posts. So we get the basic data, and we get sort of data from images. But how do we get data on social or all kinds of other interactions, and how else can we track animals? So for many years now, at least insects, people have been tracking by putting different kinds of markings and barcodes. And you may see there the ants from University of Lausanne, Keller's lab, and that's a job for an undergraduate student, of putting QR codes on every ant and then tracking them with video cameras. You can barcode your bees and flies, or you can color code your ants. Ian, thank you for your images. Or you can tattoo your frogs and track them with videos. But these are relatively small animals, and you can kind of put them in confined space. None of them are actually here, at least in the wild settings necessarily. For bigger animals, you can put GPS or radio collars, and then infer the network of interactions by, being close enough for long enough, and then maybe add orientation. And you can do that even for baboons. And that's 2012, right? Right, yes. 2012 tracking data. There's Meg putting a color and checking the vital signs. And that's the network you get out of it. So here's a day in the life of a population of baboons that was tracked attempted for a month. We have good data for about 2 weeks at GPS, 1 Hertz resolution GPS, and at 30 Hertz accelerometer. But this is GPS data. So they wake up from in the morning, from where they spend the night on this, over the river, on the trees, and then have to cross this open space and watch it. Okay. And then eventually get to the other side to eat. So I have a question for you, and those who know the answer, you're not allowed to think even. Watch the what's going on. So there's the first, you see this first group of animals kind of starting, and right, so there they are going. Now there's another one that's going to kind of follow. Watch that one that's following. Now coming back, something happens, and then continues. So as you're watching this again, the question, that one that joins the first group comes back and so on. Juvenile or adult, male or female, dominant or subordinate. So what's the status? Who is that individual? What do you think? Tell me a story that you think is happening there. That's the quiz part. So, right, so there's the first group. They're going. And then there it is. One more joining. Coming back. Something happens. Okay, so you can use your yes, no voting function in the expressions on Zoom. Male or female, if you think that this is a male, yes, female, no. Let's see. You can vote. All right, well, that's the male, okay. You can also use the yes, no, by the way. In that expressions, there is no no. So some of you think this is a male, okay. Juvenile or adult? So if you think this is an adult, yes. A juvenile, no. No? No? Okay, one person is voting. Right. And finally, dominant or subordinate. If you think it's subordinate, yes. Dominant, no. So now that you've had some time to think about it, and Meg can correct me if I'm wrong, that was an alpha male. And the thing is, a lot of people, think because we draw the network that somehow there is this juvenile that's the story I've heard told me, like tries to go after the first ones, bring the rest of the population with them. That's not what's going on. This is an alpha male that really sort of waits for the first group to go, okay, they're still alive, and then comes back to assert his dominance on the rest of the population. And what happens there when the network kind of bounces, it's that the population scatters. By drawing the network, we fool you. We fool ourselves to thinking that somehow there is cohesion, that somehow he's trying to bring the rest of the population with him. And that's the danger of drawing a network when there is maybe the wrong network. And the other aspect of it is also we're not even getting the information on beyond their locations of what it is that they're doing. So we're not getting this data on actual social interactions. And so we attempted to augment the collection, the data collection, by adding boon bits. So this kind of the bracelets that collect more than just location. That's still ongoing. We're still trying, but again, that's a job for graduate students and postdocs to test all of these things. So that with that information, we can really have a richer set of understanding of data of who is doing what. And of course, you test it not only on graduate students, first on graduate students, but then also an animal that looks most like baboon, but you can actually easily handle, which turns out to be a sheep. And there is a bracelet on her left front foot. So then you have to translate that data into something which is actually biologically meaningful, right? So you can get another computer science graduate student. There's Guido looking at Grace's color and trying to translate it directly into something biologically meaningful. Or you can use machine learning to go from the sensor data to ethograms of behavior, different behavior through various approaches, including most recently attention networks and recurrent networks to really extract that information of behavior semantics. So then we have translated data to behavior, at least. Now the question is, how do we go from individual behavior and information about individual, maybe co-occurrences of these two have been seen together at the same time, or maybe even interacting, to actual network structure? Because as Damien has pointed out in that networks, the depending are very fragile models depending on how you define networks. So for example, in this case, we can say, okay, my three nearest neighbors, my network is my three nearest neighbors, or anybody within this the radius of particular radius. That completely changes the definition and the topology of the network. Even when we say close enough for long enough, there are different ways of saying what's close and what's long, and depending on those, we get different networks and different topologies and different answers from the analysis on this. And it's been written, shame on computer scientists, that the first kind of danger, the alarm was raised for the first time but in biology of the dangers of just drawing networks and then taking them that the network is a network is a network. Because the network is not a network in the network. It really is a model. And a model is only as good as the information that it allows us to, the analysis that it allows us to perform. And in fact, the point of going from data to network structure and the way that the networks are defined really are as varied as the fields where network analysis is used. And we have a big survey paper on network inference. In this process, particularly in this process, scientific insight and scientific inference of going from data to hypothesis through the noticing patterns and create of the underlying philosophy, at least the structure of underlying data, really network fit is just another model. And so it really has to be dependent on the question we're trying to ask, on the relation to patterns that we're trying to understand from. And we showed that that really this task-focused network inference is the approach. Start with the task and the data and up the best of the set of structures and network definitions that best answer that question. In that case, you really looking at the network as a model selection, higher mechanism of yeah. Oh, sorry. Right. For the entire mechanism, well-developed mechanism of model selection, the statistics and statistical analysis. And the thing is that if you choose network structure, you can get completely wrong answers on the same, asking different. So we show, for example, that if you on the same data, if you're interested about label inference for entities in this network, figuring out the demographics and sex and status, versus if you're trying to predict a a connection, those are completely two different networks. And in fact, to make it even more explicit in this context of animal behavior, so this kind of approach allows is also to continuum, the possibility that there is no ability to ask, is there a network in the first place? All the possible networks that you are that then now compare and find the right network for the task. Network is the absence of network, right? The connections, the singletons, which means that the whatever you're trying to extract, the predict, is purely purely based on the autocorrelation on the aspects or the characteristics of the node itself. And the other approach is that it's the network of fully connected network, right? The other extreme of that range, that it's a fully connected network, so everybody's connected to everybody and everybody is the same. So that's kind of more of a collective behavior approach where everybody essentially is using the same model, the same, every agent is the same. And so, and you can have all the topologies in between. So here's an example where essentially there is no network, and that's work by most people present here. So Damien, Ari, Ian, and Meg, looking at what drives the positioning of an individual within a group. And really, this is the answer where it's not the network, it's the personal preferences and characteristics of an individual that end up being kind of the determining factor. On the other hand, if you're trying to predict the position, the XY coordinates of an individual baboon, then it is a network. And in fact, it really depends on where, or how far into the future you're trying to predict the XY coordinates. So this is label inference. We're trying to do label inference of a node, of a baboon in this case, and in fact, two labels, XY. And you know, you can ask, is it just past history? Does pure autocorrelation predict enough of the position of a baboon? So here's to remind you, here's that same day in the life of a baboon, and we're just trying to see where they're going to be in the future. So if you're trying to predict, there are many networks that you can define here. So there is a network of, you know, proximity that you saw before, of just being close enough for long enough, like right now. Who is next to me. There's also the network of long-term affiliates, and there are many other networks, and both of those are parameterized, and you can sort of figure out for the question that we're asking, which network structure is most predictive, right? Performs best to predict the location of each baboon. This is not a causal. I want to make it absolutely clear. This is not causal inference. This is correlation in a predictive setting, forecasting, if in the scientific domain. So by evaluating those two different structures, it can give us a testable hypothesis, or at least a hypothesis. And so it turns out, if you want to predict the location of a baboon, the XY coordinate of a baboon about 10 minutes into the future, it is really the network of closest 4 to 6 closest nearest neighbors. But if you looking about 2 hours into the future, then it is the network of most frequent affiliates, right? So for computer scientists in the audience, if you're deciding to go, imagine the reality pre-COVID when you did go out for dinner. So if you're deciding to go where you're going to go for dinner tonight, you are going to make that decision with your friends and just agree on a place. So with your frequent affiliate associates. On the way there, if you're going in a group and you have to, you know, make a decision to go on the left or right of a bush, you're just going to go with whoever is next to you. Kind of intuition. Whether that's what's going on or not, that's up to the scientists, and please ask questions of Megan, Ian, and Ari and Damien. But that's the pattern, right? That we're seeing, and that's the pattern that computational approach extracts and propose and perhaps suggest a hypothesis that there are two different timescales of decisions, and there are two different mechanisms of decisions that are happening at those different timescales. And we extracted by looking at what is the best network that predicts this at this timescale. And notice that it's not the entire population of 28 baboons, but 4 to 6 is where it really peaks. So there's something else that's going on there, that it's not just the more you add, the more information you add, the better it is, the better predictive power. So that's that kind of the then translates into some insight. So the next example and the last example I would like to give you is about, so how do we go from individual, so we have the data, we have the patterns and structure, we maybe even now have the right network at the right time scale. So how does it all sort of come together for decision making? Right? How do we really get from individual behavior to a group, the entire group doing something, changing status, going from sitting there in the morning on waking up to deciding to move and cross the open space and get to the other side, or deciding, the entire group deciding to go left versus right or change their direction. So you can again ask the esteemed group of scientists present here, and turns out in baboons at least that there's really shared decision making that drives this collective behavior. So they have a form of democracy, which is maybe no, but some form of shared decision making where it is a form of kind of majority-ish of and not that all animals are equal, but some are more equal than others. But how do we extract these kinds of structures and insights directly from data? Can we at all? And so we set out on a quest of figuring out how do we really understand decision making at the group level. So that is, by the way, the joy of working with sort of in this interdisciplinary field. It's the about a year or two of translation, because when Meg came and we started talking about how do animals how do social animals make group decisions, we had to pretty much figure out how to define every word of that sentence. You know, what is a decision? What is social animals? What is group? What do we mean make decisions, and so on? And so here's how one way of translating that statement of social animals making group decisions. So when in this favorite examples of baboons, sort of a group of baboons. So one decision, one can argue that if you look, they're first kind of milling around and just still hanging out, waking up on the trees, but then there was a decision to start moving. And the group eventually went from hanging out on these trees to the entire group started moving. The other set of decisions that came along the way is which route to take, right? To go here versus there. So focusing on, because we have data, right? And that's where we can look carefully on the data, is in the context, decisions in the context of movement. You can, the group decision means the entire group changed status. So we have to somehow find periods when the group, everybody in the group, changed state from maybe not moving from one type of behavior to another, or from direction of movement and so on. And so, but by the time the entire group is doing something, the decision has been made. So when the entire group is coordinated, the decision by that point is made. So the process of making decision comes to pre-coordination, right? Right before. So this pre-coordination interval, before the group is completely coordinated, is that process of decision making. And so we created this framework. So Chai Amarbanchadbe, who's a PhD student, who's now graduated, the whole framework of going from multidimensional time series data, which is movement coordinates of a troop of baboons, to defining the notion of following network, defining the notion of following network, where one baboon or one individual is, or one time series follows another if they have reasonably similar patterns, local patterns of the time series have reasonably similar local patterns with a slight shift, time shift. And so baboon B follows baboon A if the trajectory is similar with a slight time delay, right? So B repeats what A did with a delay. And so you can now draw a network, a directed network of who follows whom. And the thing to notice is that by the time the whole group is coordinated, right, when the whole group actually changed their behavior to the same type of, they're all doing the same thing, at that point, everybody is following everybody, right? So that network is going to be very dense, the network of following. But before that, the right before, when the group is in the process of coalescing and changing that behavior, then the network really goes from maybe, you know, one baboon is following another, but then gradually more and more and more and more individuals start following and joining this network of following, and then eventually everybody is doing the same thing. So the network of following is densifying. And so looking at this density of a following network, you can find the period of coordination, you can find the pre-coordination period, and then you can look at the many different aspects of individuals in this process. And one of the things to look at is really PageRank, which is in 1930s, it was created as a measure of figuring out who are the individuals who are followed by many, who are followed by many. So this is not new to World Wide Web and search engines, but it really is a measure that was created specifically for this person of identifying essentially leaders who are not necessarily at the front of the group or the first ones to do something, but they're the ones who are followed by many. And you notice that here that you can start figuring out that the leaders and the initiators, maybe not the leaders, but initiators of collective behavior, of coordinated behavior, as in this case, are not necessarily the ones who end up at the front of the group. So the black individual, the black circle, are not necessarily the ones who end up at the front of the group once the group has coalesced. And you can also start looking at different other aspects of group group coordination and distinguish sort of and different parameters. By tracking different parameters of the entire group, differentiate between different models of coordination, different models of the group going from one state to another and making this coalescing into coordinated behavior. So maybe it's random, just I'm going to do my random thing or follow a random individual at any given time. Maybe it's dictatorship model of everybody's following one baboon and doing whatever that one is doing. Maybe it's a hierarchical structure. I'm going to follow Meg. Meg's going to follow Carter, who's going to follow Ari, and so on. Or maybe it is I'm going to look what the majority of my nearest neighbor is kind of doing. And turns out that when you look at these parameters and look at different models, baboons fall very much into this type of coordination where it is linear threshold model, meaning that the majority of what those around me are doing. And that kind of like, whew, you know, we actually, with all of that, just kind of confirmed what biologists already discovered, but that allows us to formalize this process and look at other other questions and look at other populations and other modes of behavior and decision making. And so I've created more general abstract framework, which actually extracts patterns and provides application insight. And so hopefully I showed you examples of going in this process from data to computational abstractions that find patterns and back to application insight and scientific hypothesis, and allowing scientists really to look at more things more carefully. So with that, this is my own social network, a network of collaboration, and the funders that allow me to have fun and ask these questions. So thank you very much.
One thing, in biology, there's a really strong push for making tools and things accessible after even after having published a paper. And I was wondering how if that's being mirrored also in computer science.
Yeah, there is a very strong push, and actually for computer science. So we publish in conferences in computer science, and hopefully train the rest of the world now to recognize that this is a legitimate way to publish. But for major computer science conferences, there is now a special check that you have to, are the tools available, are the results reproducible and accessible? And so at least that is driving the culture, but particularly sort of for us, for myself, from my lab, Wildbook and WildMe was one of the big outcomes of that we're open source. So having a nice class project or a PhD of identifying individual animals in a publication in computer science conference is great, but it's not usable by many. And so starting a nonprofit was our answer to that, and we're open source, open collaboration, protected data, so we cannot open data there. But Wildbook is completely open source. There is now a community in a community forum, and people are adding not only we, but other people are adding functionality to it. Everything that we do in the lab is available through GitHub, and we're actually porting some of the more useful stuff to Zenodo, which is a platform where you can share code. The problem with, and it's a problem that is now being actively discussed in computer science community, is that for machine learning methods, there is no good way to share models. So you need to share not only code, but which data you trained on your models, what are the hyperparameter settings for your models, and what are all the conditions that were trained, and what they're now starting to be called nutritional labels for data. So what are the data characteristics, so biases. Some kind of some terms that are being tossed around are things like model commons, model zoo. We're only at the beginning, unfortunately, of that process. It's still going to take a while for us to figure out how to properly share machine learning models and methodologies, but I'm at least hopeful that we're talking about it.
You mentioned networks being models, but potentially deceiving models at times. And I was just wondering, kind of when you're using empirical data, you don't know kind of what the true network is. Mm-hmm. What's the process you would go through? I'm sure it's not an easy answer, but what's the process you would kind of think through to evaluate, is this model even beginning to get at the underlying biological questions that I'm interested in?
Right, so you're asking, great question, Carter. They're asking two questions there. Because one is, how do you figure out the most useful network for what you're trying to do? And the other one is, what network is biologically meaningful? So those can be two different networks. So really, very few times when we're explicitly, we or baboons or other animals are walking around with any kind of stick, 6 feet at this point, saying like, we're interacting, right? In this context, we are connected. So for the most part, these connections are implicit. And in fact, for the most part, these connections are just abstractions of some behavior, which is dyadic or pairwise or more so. And so really, there is no network per se, right? There's just a good abstraction that helps you answer the question that you're trying to answer. And so in that sense, network is a useful model that really allows you, it's at this point, one of the very, very few models and few abstractions that allow us to do, to look at emergent phenomena, that allow us to define something at individual and pairwise level, but answer questions and provide analysis at the entire group level. And so as such, really the first part of it, first part of the answer is, what is a useful network for the question that you're trying to answer? Because there are many useful networks for different questions on the same data. So the first part of it is a sanity check. So if you have, for the question you're trying to answer, networks are not, in general, computational approaches are really not good at answering biological or scientific questions. They're good at extracting patterns. So in that sense, network is just as good as a regression on data, right? So it suggests a relationship. It suggests a model of, or a pattern. So when you're putting linear regression, you're optimizing and you're fitting that line to optimize for the distance of your point. So you have some ground truth there, right? You have those points that you're then, that allow you to measure the fit of your regression. Similarly, for network, we're using some parameter to measure the fit of the network for that specific question. So for example, if the question is predicting the location, if the question is sort of how do animals make movement decisions, the very least the data that you have is XY coordinates, and the expression of a movement decision is the position of an individual. So at the very least, a useful network should be able to be predictive of that position. Right? If you're trying to, the XY coordinate, if you're trying to extract a network and really going back, we probably should redo. If you're asking a question of position of an individual within a group, within the entire group, really that's the label you should be predicting, right? Interior, semi-interior, exterior, front, back, whatever. So really we should find a network that's most predictive of that. And using some prior data and ground truth to verify the fit of that network to the data and the ability of it to predict in the future. So the thing is, though, unlike, let's say, linear regression that really has only two parameters, networks have many more parameters, and so you need more data to find a good fit. And unfortunately, a lot of the time we're trying to fit networks to very, very low, sparse, and noisy data. So the fit itself is not great. And we're getting like very wide range of possibly, maybe. And when you move to machine learning models, which are really just nonlinear regressions on steroids, with often millions of parameters, then you need that much more data to make anything useful.
Okay, thanks so much.
So, but if you can get more data, you can look at it more carefully.
Tanya, so I was curious to hear you talk a little bit more about your experiences trained as a computer scientist, but working intimately with biologists, about the differences in how computer scientists versus biologists, people with those trainings, approach answering questions, and what you think each field brings to the other.
Great question. What do you think? So really, you know, this, to me, it is a translation process which is the most exciting, and it is really a different way of looking at the world, and I think with the fundamental differences start at, well, they start at the vocabulary, and I'm told, and that's a fun part to align that vocabulary. By the way, the word that's considered to have most meaning across different disciplines, anybody knows? Projection. So, you know, the geometric projection, mathematical projection, economic projection, statistical projection, voice projection, psychological projection, film projection, so theater, all of these have very, very different meanings of the words. So in interdisciplinary, but in my experience between biologists and computer scientists, the word that I found with most frustratingly trying to define is optimal. We mean very, very different things when we say optimal. And if you want to really get computer scientists all riled up, try to use it in a different meaning that we really understand. Excuse me, the meaning of the word optimal. We define it. We define whole algorithms that find optimal solutions. So yes, the culture also, sort of this combination of the culture, quite often when any scientist, it's not necessarily a biologist, but biology, in fact, molecular biology was the first discipline that really started working with computer scientists, right? And so computational biology, starting with the human genome sequencing, set really a good example of how, you know, the two disciplines coming together can produce something that neither on its own can do. And but quite often, and in the beginning, even in molecular biology, the conversations between, you know, a lot of scientists see computer scientists as pet programmers, and the conversation starts with, can you write a code that does X? And the answer is no. What's your question, right? And similarly, computer scientists see the rest of the world as just data collectors, and the conversation says, can I have your data? Right? And the answer is no. What's your question? It is only when we really figure out how we ask the questions, and how different disciplines ask the same question, or a question that goes towards the same direction in a different way, and how we answer them in a different way, and in complementary, that we can do something interesting. And so really in science, it is about understanding phenomena of why and how. Right, and the computational approaches are really about the ability to extract patterns, both very rare patterns. So humans are not great, by the way, at seeing verifiably significant, repeatable, meaningful patterns from massive, noisy data. Computational approaches and mathematical approaches will extract patterns. Now, whether meaningful or not, biologically or scientifically, that's a whole other matter. But you know, if a human stares at a collection of random points for a while, we start seeing face patterns, right, everywhere, because we're wired that way. We overestimate the significance of rare events. We can't count past 7 or keep track of the things past 7 or 11 for some. So there are lots of just pure human biological limitations, and what kinds of patterns we can extract out of massive amounts of data, right? What can we look at and look carefully? So computational approaches are good at that. We can extract, we can fit, nonlinear regression to millions of data points and find find a good fit. We also can do optimal, and I will fight you for it, for a good definition of optimal, right? We can find optimal solution. We can find patterns that maximize or minimize a well-defined quantity. And but sort of translating the biological question, the scientific question of why and how. Why is this phenomena? What is causing it? What is the connection between this happening here and that happening over there, into something that can be answered by sort of either optimizing a quantity or fitting or extracting repeated patterns and looking at the repeated patterns in the context of all the other possible patterns. That is where sort of once you have that translation, and the thing is, we quite often, what ends up being, and that's the joy, is that we can make a good argument for the translation from biological question to computational. And so if we answer that computational question, yes, we can provide insight for biological hypotheses. The problem is quite often computational questions end up being too difficult. So we start approximating, we start producing sort of near optimal answers, and that's where the connection is lost. And so this is for, I don't know if there's still computer scientists left in the audience. Almost every interesting question is computationally infeasible, turns out. So if you can argue that, yes, the optimal solution will answer that biological question, we're optimizing for quantity, not for the topology of the solution, not for the structure of the solution. So near optimal solutions quite often have very, very different structure, right? And but biologically, you want the structure, not the quantity, the pattern, underlying pattern that has that measurable quantity. And so that's where, and I don't know if that is very abstract, but even in sort of in molecular biology, when you want to do sequence alignment or behavior, let's go back to behavior and network analysis. So quite often, we talk about initiators, and we one of the models that we considered also is linear threshold or cascade models. So there are models of behavior propagation in the network, and you can often ask, and we ask, who are the most influential individuals? Influence propagation, find me the best 3 individuals who, if I start with them, they're the ones who will propagate the behavior to the most network, right? So because and one can maybe argue that if you indeed find repeatedly those 3 individuals, then they are the 3 most influential individuals in this population. But the problem is that we can't actually solve that problem optimally, computationally. We can find an approximation. So yeah, I can find you the 3 individuals that almost propagate to the most of the network. They're not quite the best optimal, but good enough. And so the thing is, the good enough individuals might not be the 3 individuals. By optimizing the quantity and approximating the quantity of propagating to the most network, right, to the most individuals in the network, we might not actually find the best structure, which is what biologists care about, which is you want the 3 individuals, not the A3 individuals that, influence good enough number of the individuals in the network. And so that's an example where the disconnect quite often happens. And so this translation back and forth, and this is just one example where this translation back and forth and how we make assumptions and how what we optimize on and what the answer from each, what's your question and what's the answer, to me it's the fun part, but it also is where really the most attention needs to happen to really make a truly interdisciplinary research and interdisciplinary connection happen, and where one discipline happens to bring in more than.
So, well, I'm one of the computer scientists. So maybe the question goes in a different direction. I want to work on human data and not animal data. So there's a lot more patterns that are not like leader follower, but action reaction. And the reaction might, depend on the action, but not be the same action actually. So I was wondering, how far do you think your methods and this network structure would transfer to that problem?
Thank you for that question. So yes, it translates very much. So in fact, we just published a version of it, which is variable time lag Granger causality. So based following network. So instead of the original following network, which I kind of alluded to here. So it's similar pattern, but with a delay. That's for the computer scientists, dynamic time warping based approach. But that really assumes that it's a similar pattern. But if this thing causes that thing, right, that approach will not find it, but Granger causality will. So Granger causality is when you can look at two time series or more, two time series, and ask how much better can I predict the top series by adding the bottom one versus just the top one on its own, right? So with that, sort of the lift that you're getting by adding, that's the Granger causality part. Now, Granger causality, you can also ask it with a shift, with a delay, this question. And so what we just added is that the shift is in the standard framework until now, it's a fixed shift. So how do you extend it to variable length shift to find also what is the right shift? So kind of adding that, and that allows you really to look at two time series or more, where in terms of the patterns themselves don't have to be identical, don't have to be similar, don't have to be anything. It's just asking how much better are you doing in understanding this one by adding this one versus this on its own. And so extracting this correlation, it's still, even though it's the word causality is there, it's still about correlations, right? So it's correlatory relationship of time series. And the other direction to look at, which we're starting to look, is matrix profile approach by Iman Keo, and looking at time series motifs. So that's an interesting that is a whole other direction where we're now starting to look. So it's a little bit more data mining kind of pattern mining approach to time series analysis, as well as and sort of Eric Xing is another data mining kind of approach to time series. So but essentially, as is right now, is there is a version of it you can start with. What it doesn't do, and this is the full disclosure and why we need to look at other approaches, is we're looking at continuous variable time series, things like movement, and so we haven't figured out what to do with categorical variables of just labels and behavior, things like Twitter. So we cannot do it. We cannot yet, in its current form, apply to things like Twitter network or Twitter or any other sort of categorical labeled behavior.
Question about how you do science, but so you run, like, you have the most diverse lab of anybody I've ever interacted with, both in terms of where people in your lab come from, ethnically, culturally, men and women, like it's very gender diverse as well. And I was just curious both how you've accomplished that, and also what sort of special, like whether there are challenges that you feel like you've learned something about helping the rest of us overcome in terms of making labs open to and friendly for and welcoming.
Yeah, so intentionally, because I come from theoretical computer science PhD, there is no lab. I grew up as a PhD student without a lab. There's one student at a time with gaps in between. So my advisor had total of 6 PhD students in his 30-something year career. He just retired last year. So and I was his sixth and last PhD student. So this really is a very different approach. And so because I knew I would have to have a lab doing what I wanted to be doing in computational ecology, I started looking around and seeing examples of where I thought labs worked well. So, and there are great examples out there. I looked at Simon Levine's lab. I looked and Dan Rubinstein's and Joel Brown at the University of Illinois Chicago. He was a really great mentor on how to build a lab and how to bring people from different backgrounds, and kind of looking in general of how to. Right now, there's again a lot of interest and a lot of publications of how to make your lab culturally diverse, inclusive, anti-racist, and equitable, which is great, because it is time we start really explicitly training people to do that. But I mean, the secret sauce is really not there. Is really truly being open. So, the first Mayan Clahiri, who did the among other things, the first zebra recognition stripe spotter, the first zebra recognition algorithm as part of the Kenya course. His background is undergraduate in physics, and I had undergrads from all kinds of students from all kinds of different undergraduates. But it takes building a community, and that aspect of building a community and setting an example myself of really being open to ideas, and taking the time to learn and figure out of what each is talking about, whether it's biologists and computer scientists, people from a different culture, people using, often language itself is the barrier, right? So somebody speaking English with a very different accent or not very well. So taking the time to listen, but also training students and those who've gone through the lab, and Meg, you've suffered through some of that as well, of my students. They, as simple as every lab meeting, and we starts with the 2-sentence description of what it is that they're working on, and this constant training of explaining it not only within discipline but across discipline, of having constant conversations across discipline in a way that makes sense across disciplines, while being able to explain every technical detail to people who are not necessarily within the field, right? Because there are very, 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 very few people who are going to understand every single technical detail of what we do, other than the 3 people who are in your committee, maybe, and have read your your papers. So whether, you know, explaining to computer scientists, to biologists, to physicists, or natural or art history majors, it's often not that different if you really take the time to figure out, to understand what's the essence of the work. So I don't know if it's even going in that direction. A lot of the articles that I'm seeing right now, of how to make your lab inclusive. Hey Damon, bye. Good to see you. And I would love to follow up, by the way, on birds identification. Oh, David did leave. I mean, this aspect of that explicitly stating that people are welcome, shockingly, goes the long way. Yeah. The having women in the lab is, in computer science, is fortunately or unfortunately, is by being a woman. That, but I did spend and am still actively engaged in broadening participation in computing, and that work, both for women and underrepresented minoritized students, that work has to start early on. I mean, by the time they come to PhD, that's way too late. So to me, it's also a joy to work with like first graders and middle school and high school students and tell them about computer science, and particularly the work that we do, which is interdisciplinary and often appealing to a wider range of people and brings in a wider range of people into it, and then taking the time to, you know, it takes longer to work with high school students, so it takes the time, but I enjoy it. Right now I don't have a lab, by the way, so you know, all of this is theoretical. So I wanted to ask Ulrich, you also worked in interdisciplinary projects and interdisciplinary labs, including anthropology and cultural anthropology. So your experience of working in interdisciplinary, sort of across
pretty much as you said. The translation, and takes a lot of time to make people talk to each other and not forget that they have different backgrounds and don't understand everything that you say. And on the other hand, it's a great training for oneself. I don't think I would have learned as much about research in general and life in general if it wasn't like that. So I think it's very rewarding, even though it's barely countable of what's happening.
Yeah, I could have published lots more papers. But the question is, would they matter? I don't know. Martin, you're another shining example of working across boundaries and not even worrying about what the field definition actually is. I don't know if Martin is still there. Maybe not. Well, Meg, what about you?
You're not supposed to turn my own question around on me, Tanya. Yeah, I mean, I certainly think that, like, if we're talking about interdisciplinarity as opposed interdisciplinarity, that interdisciplinary work does just take more time, but it also helps you think about and see your work in totally different ways, and it's really rewarding that way. And I certainly ended up going down different lines with that baboon set that we worked on together than I ever would have gone down if we hadn't been working together on it, and ways that were really sort of productive, I think, and rewarding and exciting. And I guess in terms of thinking about, like, how do you create a lab environment where people, I think you did, whether it's interdisciplinarity or whether it's an inclusive lab environment, that willingness and investment in listening to each other and in continuing to talk to each other seems to me like a really key part of it.